Today we will be studying a topic. We will be discussing on a new topic, that is a carbohydrate digestion and absorption. Myself, Professor Neha Antal, School of Biosciences, IMS Ghaziabad, University Courses Campus. So digestion is a process which involves a basic mechanism through which complex substances get converted into a smaller or a simplest form. So it will be defined as it is the mechanical and a chemical breakdown of macromolecule or a polysaccharide into a small molecules. Basically, why do we need a process like digestion? So to run our body we need energy, and from where we get energies as our cyclic processes run. Basically, like glycolysis need glucose. So from where our body consume glucose or need glucose or take the glucose. So our body take glucose from the food which we consume. So basically, carbohydrates, there are different forms of food material which we take, like carbohydrates, proteins, and lipids, lipid food products. So the glucose particles comes from the consumption of the carbohydrate, and the form which we consume is the complex form which our body does not absorb, because our cells simply absorb the simplest form of a particle. So for this process, our body starts or takes a process, that is digestion. There are two types of digestion process, involves two methods. First is mechanical and the next one is chemical. So basically, mechanical process involves our mouth, which involves teeth. Basically mouth, which we call as oral cavity, which involves which includes our teeth, tongue, our glands, which secrete form the saliva. And the chemical digestion involves basically the role of enzymes, which convert or dissolve the food into the soluble form. So mechanical, as I have said, teeth. The function of the teeth is the mastication of the food product or convert the large particles into the smaller one, whereas the role of a tongue is to smoothen down or roll down the food particle, and the role of the saliva is first to convert the starch or complex carbohydrate into the simpler disaccharide form, and second thing, it also involves it also smoothen down the food particle so that our body or our buccal cavity easily transfer it to the esophagus part. So basically, digestion process involves our digestive system, and our body digestive system involves or made up of two different types of parts. First, we termed as a basic parts, which play a main role in digestion of the food product, and the second thing is accessory glands, where their side products involve basically the enzymes helps in digestion and dissolving the food products. So our digestive system involves, first is mouth, which involves palate, bulla, tongue, and teeth. Then next, the food goes to the esophagus. Esophagus connects to the stomach. Stomach connects to the small intestine, and then the small intestine connects to the large intestine. So these are the major organs which participate in which make up the digestive system, whereas accessory glands are salivary glands, which involves three types of different glands: sublingual, submandibular, and a parotid gland. Another one is the liver, which is the major organ to reserve the glucose in the form of the glycogen. Gallbladder, which secretes bile juice. and the pancreas, which secretes the enzymes bicarbonatides, which plays an important role in digestion in the small intestine, and the last one is the anus part. So now we will be starting with the basically carbohydrate digestion, the food which we will be taking in the form of a carbohydrate, how it will be converted into the simplest form. So basically the basic products which we consume in the form of a carbohydrates, we can include like complex carbohydrates, disaccharides, like lactose, sucrose, fructose. In the form, if I say starch in the form of whole grains, or we can say different types of not pulses, potatoes, sweet potato, normal potato, etc. And then next come to the principal side. As we have three forms of food: carbohydrate, proteins, and lipids. So there are the three types of foods have different digesting site. So carbohydrate digestion takes place in the mouth and then the intestinal lumen. Digestion is a rapid process which is catalyzed by the enzymes, and those enzymes are termed as basically glycosidases, because they hydrolyzes the glycosidic bond present between the polysaccharide chain. And the final product which we receive in the form of monosaccharide and the disaccharide. In monosaccharide we can consider glucose, and the disaccharide we can consider galactose and fructose. So these are the major these are the structures of the carbohydrate, which basically consist of three main groups. This is the example of the monosaccharide, which basically glucose. This is a disaccharide where two glucose units are joined together. This is a maltose structure, and this is a polysaccharide. It is a starch polysaccharide chain. So first is as I've started, the principal site of the digestion, carbohydrate digestion, is our mouth. So first our digestion of carbohydrates begins in the mouth. Why mouth? Because the saliva present in the mouth is having an enzyme which is termed as a alpha amylase, or we can also call it a ptyalin enzyme, which is produced by the secretion of the parotid gland present in the mouth. So the carbohydrate form which we have consumed basically are of two origin. Either it should be of a plant origin or a animal origin. If we are consuming any product of a plant origin, it will be having a starch which will be composed of two units, amylose and amylopectin. And in case if we are consuming carbohydrate product in form of animal origin, that it will be having a glycogen. So both are the forms of a polysaccharide and a disaccharide form. These needs to be converted into the monosaccharide form. So during pasteurization process, the salivary amylase acts on starch unit and glycogen unit and hydrolyze their random alpha 1,4 linkage bonds. Now the amylase action, the salivary alpha amylase action does not convert all the food product which is present in the mouth into the complete disaccharide or a simplest monosaccharide form, because in mouth hardly the food is there hardly for 3 minutes hardly. So it didn't get the enough time for the process. So the left out part is transferred to the stomach via esophagus, and then it will be transferred to the small intestine, and that particular part is termed as the chyme. So there are 2 points which we have to remember here, that in carbohydrates we have both alpha 1,4 linkage and beta 1,4 linkage endoglycosidases in nature, but human do not produce beta 1,4 bone. Therefore, that's the reason why we are unable to digest the cellulose product. So this is the basic chain. We have consumed the glucose molecule, monomer or polysaccharide, for example starch, by the action of salivary amylase and a pancreatic amylase. We will be receiving, or we will be getting the limit dextrins or either the maltose unit. These will be further converted into simpler form in a small intestine. Now the next part is further digestion of carbohydrate by pancreatic enzymes occur in the small intestine. Now we have learned about the mouth. The food is traveled from the buccal cavity to the esophagus, then will be travel to the stomach, and after stomach it is traveling towards the small intestine. Now we are starting here with the small intestine. Why not stomach? So the stomach, because our stomach is having high pH level, so the basic maximum enzymes, basically pancreatic and alpha amylase, does not work in highly acidic medium. That's why no digestion occurs in stomach. As soon as the food part, that is a chyme, travel towards the small intestine, the enzyme which will be coming from the pancreas, acting on the chyme product and converting it into a simplest form. So carbohydrate digestion halts temporarily in the stomach because of the high acidity, which activates the salivary amylase. When the acidic stomach content reaches small intestine, it will be neutralized by the bicarbonate ions secreted by the pancreas. In pancreas, acinar cells are responsible for the same, and then pancreatic alpha amylase continues the digestion of a starch. Now, after digestion in the small intestine, we have the food product is now transferred into the first part of the small intestine, that is duodenum. Duodenum and upper jejunum plays an important role in the final digestion of the carbohydrate product and the absorption of the food product. So final digestive processes occur primarily at the mucosal lining of the upper jejunum and include the action of several disaccharidases. Now what's the reason? The reason is our intestinal cells have small villi, and a number of enzymes are present here. Different enzymes and different actions, and they basically perform hydrolyzing action, where they break the bonds of the disaccharide molecule and convert the particular form into the simplest form. For example, if we have consumed the isomaltase product, the alpha isomaltase product, the isomaltase enzyme which is present in the intestinal villi break down the alpha 1,6 bonds present in the isomaltase and converts into a glucose molecule. Suppose if we had taken lactose as in a diet form, so lactose, to digest the lactase enzyme present in intestinal villi, where this lactase enzyme acts on the lactose product, hydrolyze their bond, and it will give the 1 unit of galactose and 1 unit of glucose. Similarly, if we have consumed the sucrose diet, sucrase enzyme is present, which acts on the sucrose and cleaves their bond and give us glucose and fructose unit. Similarly, if we have consumed maltose or maltotriose, so maltase enzyme is there, which cleaves their bond and gives us the glucose unit. Now the last part is all the products are now being converted into the simplest form. Now the next part is the simplest form which we have received, now they are being absorbed into the body, and they will be transferred to the different body parts to perform their actions. So absorption of monosaccharides is done by the intestinal mucosal cells. So here again the major role placed by the duodenum and upper jejunum. These are responsible for absorbing bulk of dietary sugars. For example, there are many monosaccharides unit which directly diffuse from one part to the another part, from apical part to the basement membrane, basal part, whereas some needs energy or a transporter molecule to cross the boundary. For example, galactose and glucose. Galactose and glucose are transported into the mucosal cells by an active energy requiring process which requires a concurrent uptake of sodium ions. Glucose unit can travel alone, but galactose unit needs a co-transport molecule along with ATPs and sodium ions also needed. So sodium ion act as a transporter molecule. Now monosaccharides which we have received after the digestion process, they leave the epithelial cell via facilitated diffusion. Now they will enter the villi capillaries, and then from their villi capillaries they will be entering into the hepatic portal vein, and from hepatic portal vein they will be circulating to the liver, where these monosaccharide units reserved as in the form of a glycogen. So basically we will be studying I will explain you this process by the help of a diagram. Let's see. So here's the process of absorption chart. So this is our intestinal lumen, and this is the brush border membrane. Basically our intestinal cells, the apical part of the intestinal cell, and this is the basal lamellar membrane. So the apical part of our cells have multiple numbers of enzymes which acts on a particular food product and convert into simpler form. For example, if we have consumed starch glycogen, so alpha amylase, which is present in salivary amylase, it will act on it, and it will convert this starch glycogen into two forms, like alpha limit dextrin or malto-oligosaccharide. Now again, this 30 and 70% alpha limit dextrin and malto-oligosaccharide again act by the pancreatic amylase, and it will be converted into the glucose unit, either 5 to 9, and malto-oligosaccharide into the either 2 or 3 units of glucose or 4 to 9 units of glucose. Now these are the simplest form, like glucose units we are saying. So glucose, to carry these glucose units, you can see here, this is a glucoamylase enzyme. It will receive this glucose unit, and there are transporters present. Transporters like these are basically channels, or we can say a gate or a vehicle which is used by the glucose to cross this intestinal lumen and transfer to the liver, blood circulation. So here you can see like glucoamylase, they have converted this glucose unit. So glucose unit, with the help of SGLT, like in name they have, like cars have different brands. Similarly here the transporter is the car which is acting as a transporter its name is SGLT, that is sodium glucose transporter channel 1. So with the help of sodium ion, this glucose will be traveling from the intestinal lumen, transferring from the apical membrane and going towards the basal membrane, from where it will be circulating into the blood. Now alpha limit dextrin release gives some G5 to G, means glucose 5 to 9 units. It will be again absorbed by the alpha dextrinase enzyme. Now we have learned also sucrose. Sucrose is hydrolyzed by the enzyme sucrase. So after hydrolyzing or after cleaving the bonds, it will give 1 unit of fructose and 1 unit of glucose. So fructose with the help of GLUT5 channel. Glucose unit can travel by the SGLT molecule transporter channel, whereas fructose needs additional channel, a different channel, that is glucose transporter unit. It is energy dependent, it will be needing energy, and it will be transferring with the help of a facilitated diffusion. So fructose will travel through the GLUT5. Again, lactose is cleaved by the lactase enzyme, and it will be giving us glucose unit and galactose unit. So glucose will be traveling, crossing the barrier, brush border membrane, with the help of SGLT1, and galactose again the same process. So this is how the complex carbohydrate or polysaccharide forms is getting converted into the glucose molecule or simplest monosaccharide unit, and how it will be transferred to the liver or a blood circulation.